Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello everybody, welcome back to another episode of the A-Level Cookbook. Today we're going to be doing AQA A-Level Biology Paper 2 from June 2017. Exercise caused an increase in heart rate. Describe the role of receptors and of the nervous system in this process. Over 25% of people that sat this exam did not get a single mark and only 10% got full marks. This is literally nearly all recall. Lots of people were describing nerve impulses as messages, signals, or information, and a lot of people just didn't know what each thing did. Now, receptors, they detect a stimulus, a sort of change. They are specific, and what they'll do is, depending on what it is, if it's a nervous receptor, it will send an impulse to something, some sort of thing in your brain or spinal cord, which then sends a response out, whether it's through a motor neuron or a sympathetic or parasympathetic neuron. Now here, the question has told you exercise leads to an increase in heart rate. It's your job to explain from this step all the way to this step. If you're exercising, you're going to be using up oxygen, but chemoreceptors cannot detect oxygen. They detect CO2 or H+, right? Which is when CO2 dissolves. So your first thing is you're going to talk about the stimulus, which leads to the response. So chemoreceptors detect an increase in CO2 or an increase in H+, or a lower pH, any one of those. You can't detect oxygen. Then what they do is remember, you've got a stimulus picked by a receptor, goes to somewhere in your CNS, down and like a nerve to some sort. So it could be a sympathetic nervous system, a parasympathetic nervous system, a motor neuron, right? To eventually the effector, whatever that might be. Now here, the question has told you we're increasing the heart rate. The control of heart rate is all to do with your sympathetic nervous system. Your sinoatrial node, if you chop somebody's heart out and left it there, it would be on its own. What's modulating or making it going faster and slower is your sympathetic and your parasympathetic nervous system. Sympathetic's fight or flight goes faster, parasympathetic slower. You always have a bit of the two going on. If you had only one or the other, you would just die. Like if you had only parasympathetic, you'd be dead and your heart would stop. So here, we need to eventually see how this can lead to a change in heart rate, and that's all to do with changing how the sinoatrial node is sending out impulses. So what happens is the chemoreceptors detect this fall in pH. They will then send impulses. You can't say signals or messages. That's terrible. Don't do that. Send impulses to the medulla, which is where all of this stuff is processed, right? It's all about breathing, and heart rate is all controlled in there and automatically done there. So it sends impulses to the medulla, which then sends impulses via the sympathetic nervous system to the effector, which is the sinoatrial node. What this does is then that increases the sinoatrial node firing rate, therefore you get an increase in heart rate. That's it. AMP-activated protein kinase, AMPK, is an enzyme that regulates a number of cellular processes. Exercise leads to activation of AMPK. Figure 1 shows one effect of the activation of AMPK during exercise. So remember, then here it says CPT1 is a channel protein that transports fatty acids into mitochondria. Using figure 1, explain the benefit of activating this during exercise. So over a third of people got no marks in this, which is just shocking. The idea is that you're supposed to use what the question's telling you. So again, exercise activates AMPK. If it activates AMPK, you're inhibiting the conversion of acetyl-CoA to this. This malonyl-CoA inhibits the transport of fatty acids into the mitochondrion. So if we're going to use more AMPK, we're going to stop this acetyl-CoA becoming this, which means we're going to get less of this. That's what the figure is telling you. Now it says here, CPT1 is a channel protein that transports fatty acids to mitochondria. Using figure 1, explain the benefit of activation of AMPK during exercise. So you need to go from every single step, from activating AMPK to exercise, and why it is a benefit. Loads of people are like, oh, the keyword is explain, and they just say absolute garbage. I don't think that's what you should be thinking. Yes, the keyword is explain, so you're not going to have to just simply say things, but you also need to explain how activating AMPK leads to better exercise in this sense. That's what the key thing I think you should underline really is. Obviously this as well, that's relevant, but you need to use all the information in the question that they have given you. Over a third of people got no marks in this, which is just shocking, because I guess they were just yapping about things that just didn't need talking about. So in this diagram, the first thing you can say is that when you're exercising, you've got more activation of AMPK, which means you've got less malonyl-CoA formed, right? Which then means you're not inhibiting this stuff as much. So if you're not inhibiting this thing, more of it's going to happen. So you get increased transport of fatty acids into the mitochondria. right? So that's the limit of which we can talk about the figure, but it says explain how doing this thing for some reason benefits exercise. You need to think about what fatty acids in a mitochondria would do for exercise. You can oxidize or you can respire, either one's fine, using the fatty acids to generate ATP. Because the mitochondria can also use other things as respiratory substances like fatty acids and amino acids too. So that's what it is. That's all it is. So now it says dengue is a serious disease that's caused by a virus. It's carried from one person by another by this mosquito. One method to try and reduce the transmission is the sterile insect technique, which involves releasing large numbers of sterile, infertile males into this habitat. These males have also been made infertile by using radiation, right? So then the question says, explain how using this could reduce the transmission of dengue, right? So it's saying this leads to low dengue, and you need to use the question and your knowledge to link it together. So if that's the case, then the question's told you that if we're doing sterile insect technique, well, first of all, they are infertile, so they cannot reproduce. So that's already one of them. But then all of these sterile ones, they've also just released large numbers of them into the habitat too. So these infertile ones, not only can they reproduce, but they're also competing with those that are fertile for like food and resources. So it's a double whammy because they're going to compete with those that are fertile and able to reproduce, but they also can't reproduce themselves. And also competing. It's just an all-time just disappearance of them. Bye-bye. Now it says, describe how the mark release recapture method can be used to determine the population at the start of this investigation. So what we need to do is we need to go and say all of the things that we do. So when you're doing a mark release recapture, what you're doing is you're collecting a sample of mosquitoes, you mark them, and you release them. Right? That's the first step. Then what you need to do is you've got to leave enough time for those ones to disperse, because otherwise, imagine here's your population, you've got all these mosquitoes here, and you just selected like this, right? You mark them, and then you slap them back in this little cluster here, and then like a day later, they haven't had time to go and balance out again, so you come and get them again. You're like, wow, there's so many of them, or like so little of them. Sorry, because obviously the formula is different, but we'll get to that. You want them to spread out and have enough time to do that. So you should always say that. That's how you do a mark release recapture by definition. Then what you do is you estimate the population size by doing number in first sample times number in second divided by the number marked in second. There you go. Then it says the release of radiation sterilized Aegypti has not been very successful in controlling the transmission of dengue. Suggest why. So again, what they've done is they've radiated them. For some reason, it's not really stopped dengue being spread, which they've told you by in the question is by being through a mosquito. Immediately, what's saying to me is that it's probably going to kill them. It's going to shorten their lifespan, which means they can't really spread it, or they don't have enough time to even try to be like, reproducing or competing with the males, right? Now it says, recently a new method was developed to control these. Scientists produced transgenic male carrying a lethal gene which kills their offspring before they could reproduce. The scientists release these transgenic males every week in one area of a city in Brazil. So one area of a city in Brazil. At regular intervals, they determine the number per kilometer squared in the area where the transgenic male were released and in a control area where none were released. Figure 2 shows the results. So always just scribble down what an independent or dependent variables are here. Now, what are they changing? Well, they're changing the area, and also they're changing whether they're transgenic or not, right? And whether they're dependent. So it's tempting to be like, oh, well, the amount of the control. It's not. It's how much they're measuring, which is the number of Aegypti per kilometer squared. It's always good to scribble this down because it might help in a question, like evaluation questions and things like that. So it says, suggest why they released more transgenic males every single week. Well, immediately, the question's told you that there's a lethal gene which kills them before they could reproduce. So, I mean, they could just probably die before they've even had a chance to do anything. So it's probably to replace mosquitoes that died. And as well as that, I guess you could say something like, for example, to maintain enough of them in numbers as well. A lot of them were just saying that they, died, but they didn't really clarify that this is because we need the numbers to stay high enough. That's what the question is looking for. The release of transgenic males proved successful in reducing the number of Aegypti. Describe how the results support this conclusion. So when you're saying that, it's always just saying, look at the results and say what's going on. So I should have done before, sorry, I always do, is always have a quick look at what's going on here. So we've got time, right? If you look at the treated area, the number of mosquitoes per unit area drops the first week, kind of goes back up, drops again, but then it spikes back up, and then starts dropping again. But that's because every single week they're adding more, so it's dropped, it come back, so drop, so it's them doing that. In a control area, they did not add any transgenic ones. So the number of normal ones went down, up, and spike up and down, 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 right? So we need to look at the data to see how it supports that this method of treating works. So if we look at the start, it's at 700, and at the very end, it's nearly like 0. So after the entire 16 weeks, the number of mosquitoes at end is lower than the control, because in the control it's just fluctuating up and down. So that's immediately a supportive thing. We should really say at what time, at what point, because it says describe the results, so like at 16 weeks-ish, for example, and there you go. You can't just say simply releasing more equals like, you know, low. You need to say that, you know, this has to be a comparison, a comparative statement. So at the 16-week mark, it starts to become lower at the very end, but it becomes really, really low beyond that, beyond like what the 12-mark point. Scientists investigate the effect of regular exercise on skeletal muscle fibers in mice. The scientists compared the muscle fibers of mice after 6 weeks of regular exercise and trained mice with those of mice that had not exercised. The scientists stained the muscle fibers from both sets to show succinic acid dehydrogenase activity, and the darker the stain, the greater of the succinic acid dehydrogenase activity. So again, dependent, independent. So what's the independent? Is control versus trained mice. So have they had exercise or not? So no exercise versus exercise, and their dependent is what they're measuring? The staining of the succinic acid in muscle fibers. So then if we have a look in the controlled mice, there's lots of little whitey bits and then some darker bits, but here there's a lot more darker bits in the trained mice versus controlled. Fine. Succinic acid dehydrogenase is an enzyme used in the Krebs cycle, suggests one reason for the difference in staining between the muscle fibers of the controlled and trained mice. In this question, around 2/3 of people did not get the mark. They just kind of said, there's more of this activity and there's more Krebs cycle, which has already been told in both of the stems of the questions. You can't just repeat back what the question says. It's telling you why this is happening. If the question says there's more of this, there must be more Krebs cycle, which means there must be more mitochondria. Or alternatively, you could say, for example, there's more aerobic respiration as well. Because you don't say just respiration, because anaerobic has no Krebs cycle in it. The scientists then compared the length of time that control mice and trained mice could carry out prolonged exercise. The trained mice were able to exercise for a longer time period than the control mice. Explain why. So here, you've got to use what the question says and then go back to what it is. Less than 10% of people got all of the marks. Lots of the answers were just general changes like more mitochondria, increases supply, but not actually explaining why on earth they can do more. So here, if they have more succinic acid dehydrogenase and more Krebs cycle going on, it means that they have more aerobic respiration happening. So if they have more aerobic respiration, they're going to have more ATP. So that means that because they're able to exercise for a longer time without having that muscle fatigue, it means that there must be less anaerobic versus the respiration and more aerobic in the exercise mice, which means that, remember, the anaerobic respiration makes lactate or lactic acid, so there's going to be less lactate. There you go. The scientists determined the mean diameter of muscle fibers in trained mice using an optical microscope. The circular area of one field of view, so the field of view area was 1.25 millimeters squared. The diameter of this area was equal to the diameter of 15 muscle fibers. So diameter was 15 muscle fibers. So then it says, using this information, calculate the mean diameter of muscle fibers in this section of tissue. Right. We need to find the diameter of this area then, and then we can find out, because that whole diameter of this area that the muscles they see in is 15 muscle fibers. So if we divide by 50, we can get what we want. So pi r squared equals 1.25, which would be the area of a circle. So r squared equals 1.25 over pi. So that means r is going to be the square root of 1.25 over pi. So d would be 2 times the square root of 1.25 over pi, because that's the diameter, right? So if I do that, we get a diameter of 1.2616 millimeters. Careful, because the area is in millimeters squared. We've just worked out the diameter, which is the length, so it's millimeters. So then, this diameter is 15 muscle fibers. So one of them is going to be this answer divided by 15. If we do that, we get 0.0841, right, millimeters. But they want it in micrometers, so you times it by 1,000. If we do that, we end up getting 84.1. So scientists also compared the diameter of samples of muscle fibers taken from young and adult mice, and some of their results are shown below. So number of muscle fibers is on the y-axis, the muscle fiber diameter is on the x. And we can see that the young mice tend to have smaller muscle fiber diameters and more muscle fibers, whereas the older ones, the adult ones, tend to have bigger diameters, but the numbers are a lot less. And as well as that, there seems to be a wider spread of results here versus here. So there's a bigger range of diameters that these older ones can have than the younger ones. So already I've done the task, haven't I, really? It says describe two differences between these samples of muscle fibers. When you're doing that, though, you've got to do like a numerical comparison, though, of either of them. So, for example, well, here, the younger have a narrower range of diameters, right? So that's going to be what, like 12 to 48 versus adults, which have a range of what? 18 to 86 micrometers. So that's one mark there, done. Now, so the next thing is to say, well, we could see here there's a peak at what, the 50, right? But that's not saying that if you have this, you get 50. What it's saying is that 50 fibers had this diameter in the young. So be careful. A lot of people, I think, were just kind of like saying that this is the mean, for example, or they were saying that these are like, you know, causing this. It's saying that 50 of the fibers, the number of them, had this diameter. So then here in young, there's a peak at 30 micrometers diameter, right? So most fibers here versus the other ones, which peaked at what, like 50 micrometers. So just be careful to read the graph carefully and understand what's going on. A student isolated chloroplast with spinach leaves and they put it in a suspension. They use chloroplast suspension and DC-PIP solution to investigate the light-dependent reaction of photosynthesis. DC-PIP is blue when it's oxidized and colorless when reduced. Remember that oxidation is losing electrons, reduction is gaining, but you can't oxidize something without reducing something else. So if you're losing electrons, it has to go somewhere else. If you're gaining electrons, it has to have come from somewhere else. And you always need to say where, from, and to. Now it says here, here's the test tubes as follows. So we've got no chloroplasts, and we've got DC-PIP in light. So there's no, it's just DC-PIP and light only, really. And the second one, we have chloroplasts plus DC-PIP, but we have no light. Right, so the only difference between these two, well, there's two differences. One has chloroplast and no light. And then the third tube is chloroplast plus DC-PIP and light. So any differences between here and here can tell us that light must be the only change, provided you do the experiment perfectly. And any difference between here and here and here and here must mean that chloroplasts are the only factor that's in difference. So I'm going to compare these two first and then these all. The student recorded the color of DC-PIP at the start, the end, and they'd be left at 20 degrees for 30 minutes, and the tables are shown below in the results. So if we look, always write where your independent and your dependent are. So your independent is going to be what? So it's DC-PIP's presence, light, or chloroplasts. And then the dependent is the color the DC-PIP is at the end. If you look at the tube, so tube 1 is blue and it's blue. So immediately that's telling us that DC-PIP and light alone is not enough. You're going to need chloroplasts, because that's what these show if you add the chloroplast and the light. Because between here and here, the only thing that's different is chloroplasts, right? So then that means we need chloroplasts. Chloroplasts to be able to do things. Now the second tube is showing there's blue and blue. So between here and here, we've added chloroplasts, but that's still not enough because there's no light here. So clearly it must be that maybe there's light that's needed. If you look here in the third one, it's going from blue to colorless, which means that between here and here, the only difference was that there was light. So we need light to actually, you know, make the DC-PIP go colorless, which they've told you in the question was when it goes colorless, it is reduced. So the solution that was used to produce the chloroplast suspension had the same water potential. Explain why it had to be the same. It's to stop osmosis, which can cause cells to shrivel. Sorry, not cells. We should say what specifically is, sorry. Chloroplasts to shrivel or burst. Now, many people just kind of said plasmolyzed, turgid. Dude, no. Just don't use weird terms that have no relevance. Turgid, no. Like plasmolyzed, whatever, fine. But like, that's for a cell. Plasmolyzed that's when it's a cell. We're not dealing with cells. We're looking at chloroplasts and turgid as well as a cell. It's so that the organelles don't shrivel or shrink. It's just safer to say shrivel or shrink than to try and be big, you know, Captain Big Nuts. Now it says, explain why they set up tube 1. So look at tube 1 and the other ones, and people just say to allow comparison. Your job is to do the comparison. That's useless. Just saying, oh, to allow comparison. What is the comparison? That's what it's asking you. So if we look at tube 1 is DC-PIP and light. Look at the other one with DC-PIP and light. What's different? So DC-PIP and light versus DC-PIP and light with chloroplast. The results show it's blue, blue, colorless. So that means having chloroplast is necessary for this to go colorless. So then the effect is 1 to show chloroplasts needed for reducing DC-PIP, but that's not enough because that's just comparing it with here and here. Now we need to just compare it with this tube as well. So we know that chloroplasts are needed, but if we have no light, nothing happens. So then it also shows as well that light has no impact on it. Because if you do that, it's blue and blue as well. So then clearly, it's just to make sure that light isn't for some reason making DC pip go funky as well. So it's just to show light doesn't affect DC pip. Lots of people would like to say allow comparison. No, it's your job to do what the comparison bit is. You need to explain why. Explain the results in tube 3. So if we look at tube 3, we've got blue and colorless. Clearly, it means that this DC pip has been reduced. So number one is you're going to say DC pip is reduced. But remember how I said before, you can't just give electrons to something without saying where it's come from, because you have to reduce, when you reduce something, something else has to be oxidized. So you need to think where those electrons came from. So it's DC pip is reduced by electrons. Those electrons are from the light-dependent reaction, or so you're going to be specific, the chlorophyll. Now, the student evaluated the effectiveness of different chemicals of weed killers by looking at their ability to stop decolorization of chloroplast suspensions. They added different concentrations of each chemical to chloroplast suspensions with DC pip, and they looked for the IC50 for each one. So here, what they're changing is the concentration, what they're measuring is the IC50, and the question's told you the IC50 is the concentration which inhibits decolorization by 50%. So you need to think in your head why on earth they're doing this experiment where they're changing the concentration of this weed killer and waiting for when it drops by 50%. Why not just do the 100? Why 50? Well, imagine you had to sit there and you had to keep adding loads and 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 loads, and you're using liters of it until you finally get to that point, right? Maybe it just might take freaking ages. So what we could do is we could just say wait till it gets to 50, and the moment it's 50, that's our endpoint. So what we're doing is it's providing an endpoint, which means we can then use that endpoint to compare different concentrations, because I know if I use this much, I'll drop it by 50 rather than waiting for the whole thing to finish. So now it says, explain how chemicals inhibiting the decolorization of pip could slow the growth of weeds. So again, read the question, please. Don't just jump into saying random garbage. It's saying here, these things are stopping decolorization of DC pip, which are effectively stopping reduction, right? So then it says, how does that slow the growth of weeds? Now it links back to your understanding of the entire point of the light-dependent reaction. The entire point of the light-dependent reaction, or non-cyclic photophosphorylation really, is to produce ATP and reduced NADP. Why? Because the Calvin cycle uses it to convert, well, first of all, IBP combines with CO2 to make a 6-carbon compound, so it becomes 2 times GP eventually, right, with Rubisco, and then that gets reduced by the reduced NADP, which gets oxidized, and it uses ATP to make TP, and that's either regenerated or makes something else. So here it's saying, how does this lead to this eventually? So what it means is that you're going to get less reduced NADP, because remember, if we're using this thing that can stop reducing of DC pip, it means we can stop reducing of NADP. So if we get less reduced NADP, that also means we're going to have less GP being reduced to TP, which means less useful organic compounds, like glucose. Many people were just saying that, like, you know, oh, less Calvin cycle happen, or less glucose made, or less photosynthesis, but they didn't actually say this specific step of what happens in the Calvin cycle as a result. So it's just really important to almost step by step describe every single process from one to the next, and don't jump steps. So arbuscular mycorrhiza fungi are fungi that grow on into the roots of the plants. AMF can increase the uptake of inorganic ions such as phosphate, so just one way an increase in the uptake of phosphate can increase plant growth. Now the word myco means fungus, rhiza, root. Fungus, root fungus, right? So that gives it away what's going on here. Now again, it says suggest in one way in which increase in the uptake of phosphate can increase plant growth. Now, you need to think in your head what the hell this plant's going to use with phosphate. So it's used to produce, you know, something that's got phosphate in, like ATP, DNA, you know, phospholipids, RUBP, TP, RNA, GP. A lot of people just said proteins. No, dude, come on. Suggest one way in which AMF may benefit from their association with plants. So only 60% of people got this, nearly. Now, it's asking you why this thing living on the roots of plants and doing its job for it means that it's going to have a good time. It's going to be living. Well, it's a mutual relationship. It's going to have a symbiotic relationship, which means it's going to have a beneficial exchange with this plant. It's you know, hey, you have some of this, bro, and you can give me some of this in return. In this case, what it must be is that, well, the plant's probably going to give it, you know, like glucose or something. Plant provides glucose or useful organic compounds like that, or amino acids, lots of different things like that. You need to give an example of it, of what the plant's giving to them. So now it says scientists investigated the effect of different AMF species on the productivity of plants in a plant community of this grassland ecosystem when growing in or on soil containing different phosphate concentrations. What they did is they did identical plots of these things containing seeds of this plant species found in the ecosystem. The scientists added different AMF species to different concentrations of phosphate particular plots, the control plots. With that, AMF species were also set up. After 20 weeks, the scientists determined the shoot biomass for each plot, and the results are shown here. So now you've got to have gonads of steel to go into this and just be like, yeah, I'm going to just go straight into this without even thinking about anything, right? Independent, dependent, all the time. It always helps. It doesn't have to be like some proper formal one, it's just an idea of what they've done. So what are they changing? They are changing whether there's AMF present, the AMF species, and its presence, but also whether it's got the phosphate concentrations changing as well. What are they measuring? They're measuring log E of the shoot biomass. Now the reason they've used a log scale is because these, if you unlog these, that would, like here, that would be e to the 1, e to the 2, and so forth, which is like 2.7 to the 1, 2.7 to the 4. You can see that gets very, very high, very fast. So because there's a big range of results, they're going to try and squeeze it on here like that. So it says, explain why an increase in shoot biomass can be taken as a measurement of net primary productivity. Now, net primary productivity is the amount that is available to the next trophic level. Primary means that this is a plant for converting sunlight into chemical energy. NPP is GPP minus whatever a plant's losing, which is its respiratory losses, because plants still respire. The question's saying, though, how you can measure increased shoot biomass and use that as a measurement of that. The biomass represents dry mass of carbon, right? That's why you're able to do that part. Now the next part is to explain why the shoot biomass specifically is useful, right? And why it can be net primary productivity. So this means it can represent this exact thing here. So it's your gross primary productivity, you'd have to write the whole thing obviously, minus R. Because it's not going to be, you know, some of that will have been lost by respiration. If we're taking a shoot and measuring it like that, then, you know, we're not going to be able to factor in the respiratory losses. They're gone. It's finished. Finito. It's not there anymore. So now it says, using this data in figure 5, evaluate the effect of plant productivity of adding AMF species and adding phosphate to the soil. So whenever you're doing this kind of thing, you need to think of what a conclusion is here in this question, or make, you know, kind of go with one. And then here, look at what's supportive of that statement, what's contradicting, and then issues with validity, right? This is the one that everyone fumbles horrendously. Because what people say is they'll just say some sort of rote-learned answer of other factors, and that and that. And then they'll be like, I don't know, results due to chance or something, without saying any useful stuff. Here, what you need to say is look at the data, what's supporting whatever the statement is, what's directly going against it, and explain why or what could be going on. And validity issues are things like, can you take this and apply it to every single plant in the universe? Can you apply it to every single scenario? Can anyone else do this in the world and they'll get the same thing? Which means you'd go through step by step of their method, step by step of what the results show, what did they measure, what are they relating that measurement to, and is it representative? For example, if I get a room full of people with chocolate, and there's 10 of them, and I make them eat chocolate, and they all shit themselves, and I come up to you and say, dude, I did this experiment, my method involves selecting 10 people, I gave them chocolate, and they all shat themselves. Therefore, every single person in the universe who eats chocolate will shit themselves, and it must be because of the lactose in it, and they must all be lactose intolerant. You would not believe me, because immediately you would be like, what the hell are you talking about? 10 people, and you gave them chocolate, and they all shat themselves, and like, now you're saying they have lactose intolerance, right? When you're evaluating, you're looking at what's supporting that and contradicting, and then validity problems. So here, in my supportive statement, yes, 10 of my 10 people shat themselves while eating chocolate. That does suggest every single person in the universe would. Contradicting statements, there are none, because all 10 of them shat themselves, right? The validity issues are mostly what my experiment is. There's only 10 people, small sample size. I'm assuming that because they shat themselves because of the chocolate, that immediately means they have lactose intolerance, which is not necessarily true, because they might not have, you know, some chocolate might not have lactose in it. There might be varying amounts. As well as that, we don't know what their age, sex, whether they have any allergies or history of problems like this. Also, as well as that, we don't know what else they've been eating. We don't know if it was only chocolate they ate. We don't know how long it's been. Those are all issues to validity as well. And there's no control. There's no comparison. I just have 10 people and they all just ate chocolate and shat themselves. Who am I comparing them against? There's no one in that group who had no chocolate or a placebo with no lactose in. So that's how you tackle those. So here, we need to look at what's supportive of the statement really being that there's phosphate affecting plant productivity. So phosphate, and AMF affect plant productivity, right? So if you look at what's supporting this statement, let's do it bit by bit. So if we're adding AMF first, so there's no AMF, it's a control. If there's no AMF, there's barely any difference, there's a slight increase here. So I mean already that's kind of a contradicting thing because there's barely any increase. So contradicting, adding phosphate alone, to soil alone has a very little increase in biomass, right? Here and here, there's barely anything. Now let's do adding AMF. Sorry, I diverged there, so now let's do adding AMF. So if we add AMF, which is all these three, remember? Well, there's some going down here, there's some going down, but we're looking for supportive statements. So when you're adding this species and this, it goes up in both circumstances. So adding AMF, so you'd add, say, adding this scutella blah blah blah, and the intro blah blah blah, increases the shoot biomass, right? And also, we're still not done because we're still looking at adding phosphate. We're looking for anything that supports it really here, right? So this is adding phosphate only, but if we're adding phosphate and also doing this, if you look at glomus here and we add phosphate, adding phosphate makes glomus even higher, which is even more like, you know, of a supportive point towards phosphate. However, if we add phosphate to the whatever, it goes down. So actually adding phosphate to blah 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 actually makes the biomass go down, which is a contradicting statement. Can you see how I've rinsed all of these points out just by looking at that when you're evaluating it, comparing one by one, seeing what's happening? Now, issues with validity. So can we go and do this and apply this to every single thing? Can anyone else in the universe get the exact same results? What's the problem with saying that adding phosphate or AMF can affect plant productivity? So let's go through each step one by one and see what they did. So they got, They got productivity of the plant and community of grass, prairie land, ecosystem growing in a soil for phosphate concentrations. They set up identical plots of this containing seeds of the plant. They added different AMF species, different concentrations of phosphate. So already it's looking pretty well controlled and nice. So not really helpful there. I can't really talk shit on that. But then if we look at the soils that's got different phosphate concentrations, fine, whatever. The scientists, what else did they do? The control plots of that then were done. After 20 weeks, the scientists determined the shoot biomass for each plot. So already one thing is that it's only been 20 weeks. We don't know if beyond that 20-week marks it could actually go even higher. Maybe they measured it too early. So don't know if measured too early. Then as well as that, these old bars that are showing differences in the shoot biomass and the means, are they means? Oh, the shoot biomass for each part. So even means we have no statistical tests. I don't know if this is actually higher or lower. I don't know if any of these are higher or lower statistically significant. So there's no stats tests. So therefore, you need to say why that's important. Don't know if it's significant or due to chance. You can't just jump in and say, oh, no stats test, and you can't say other factors. The amount of people go read an AQA like A-level biology examiner report. Every single one of them at some point will say students just said rote learned answers including other factors and did not quantify this. They're so mad. You guys are annoying them so much with this other factors garbage. If you're going to say other factors, you need to say what it is. Using the E2X butter calculator to calculate determine the rate of shoot biomass production in grams per day for the control plot with normal phosphate concentration. So if we go here, normal phosphate concentration is the soil plot, right? So in this case, we have got what? 2.0, 2.4? So the log E value is 2.4. So then the actual amount, it's shoot biomass production, is going to undo a log, you would do E to the log value, right? So in this case, it's going to be E to the 2.4, which gives us 11.023, right? Now the thing is here though, this is the shoot biomass after 20 weeks. So in order to work out the production in grams per day, you would do your answer 11.02 divided by, this is per day, so it's going to be divided by 20 times 7. So we do that, we get 0.0787, which is roughly 0.08. Keep it to the same number of significant figures in the question if possible. Each year, a few people with type 1 diabetes are given a pancreas transplant. They are not used to treat type 2 diabetes. Give one reason why they are not used in type 2 diabetes. So the problem with type 1 diabetes is your immune system destroys your islets of Langerhans, the beta cells specifically, so they don't make any insulin. So treating them involves giving them insulin or something that can produce insulin. Type 2, what happens is your insulin receptors are no longer responsive or less responsive to insulin. So your body's still making insulin, but it's not actually responding, it's not working, and that's because they can be damaged or faulty or things like that. So here, why is it not used in this? Well, it's because type 2 diabetes, they still make insulin. The issue is, however, is that they're just less responsive to it, which actually is the second marking point here, really. I should have put that there, whoops. That should be there. Another point you could say is that type 2 is instead treated with diet by exercise or other diabetic drugs, but like, you know? The pancreas produces a hormone insulin, state which describes incorrectly the action of it. So insulin is all about shoving glucose into cells, as in making it go lower. So whatever processes will decrease blood glucose concentration will be the insulin one. So if we have a look, Activates enzymes inversion of the conversion of glucose to glycogen. Yes, because if you're putting glucose into glycogen, you're decreasing it in the blood. So that one's correct, but they're looking for incorrect. So that one's out. Correct. Controls the uptake of glucose by regulating the inclusion of channel proteins in the surface membrane of target cells. Again, that is correct, because if you have channel proteins in a cell, they're able to take that glucose in and take it away from the blood. So that one's out. Attaches to receptors on the surface of target cells. That's how all hormones generally work that are like insulin, except steroid hormones, which can go into the cell. So that one's out. Activates enzymes in the conversion of glycerol to glucose. Well, that's converting something that isn't glucose into glucose. We're making glucose go higher, which is immediately wrong, because that would increase blood glucose concentration. When you're converting something that isn't glucose into glucose, you're making new glucose, and that's called gluconeogenesis. Right? Glucose, new, genesis, making. It's in the name. You are making new glucose out of something that wasn't glucose. Glycogenesis, here, glycogen, here, genesis, making. So you're making glycogen. Glycogenolysis, so it's glycogen. Lysis means breaking down. So glycogenolysis is breaking down glycogen. What other ones are there? Glucagon, the hormone. Glucose, agon is providing or increasing amount of. So provider. So what it's doing is it's making glucose go up. Insulin, putting something in, away, in, right? What other ones are there? There's glycolysis as well. So glycolysis. Glycus, sugar, glucose, lysis, breakdown. So those are the words that you'll come across. It doesn't really make sense to say glucosolysis. That just sounds a bit weird, so that's why we call it glycolysis for respiration. So scientists investigated the use of induced pluripotent cells, IPS, in type 1 diabetes. What they did is they used 4 transcription factors to reprogram skin cells to form IPS cells, right? So, oh my god, I can't underline on this dang whiteboard, dude. It's so out of sync. The scientists then stimulated the in vitro differentiation of these IPS cells to pancreatic cells. So what they did is they did 3 experimental groups. So we got type 1 diabetes with IPS cells. So type 1 plus IPS. Number 2 is type 1 diabetes untreated. So type 1 only, no IPS. And then here, 30 minutes without diabetes with untreated. So it's no type 1, no IPS. Don't just sit and say 2-hour comparison. If it ever comes up, you need to say what the actual comparison is. If it's saying, why did they do these tubes, you can't just say, to allow the comparison. To say, this is the effect of this or whatever. The whole point is type 1 plus IPS versus type 1 without IPS means that any changes we see between these is ideally because of just IPS being added. Between here and here, no IPS, but type 1 versus no type 1. Any changes between these two must be because of the type 1 if we do the experiment right, if it's a valid experiment. The scientists measured the blood glucose concentration of all 3 mice on a weekly basis for 12 weeks, and here's the result. So we don't go in without just being like, okay, I'm now going to not look at the figure and absolutely jump to the question and try and say garbage. Look at what's going on. Group B, remember, is type 1 diabetes, no treatment, so you can see that their glucose is going up as expected because they got type 1 diabetes. Group A is they have type 1 and they have IPS. They start and it goes down and it kind of stays and it's level around the point at which normal mice without any diabetes are the same. So it's showing that there's some suggesting, helpful treatment here. It says here, suggest how transcription factors can reprogram cells to form IPS cells. You can't just say genes are expressed, inhibited, switched on or off. You need to say how this will lead to them being switched on or off. So transcription factors are things that can bind to DNA promoter regions on the DNA. So it's a sequence that says, hey, let's get going. And they can either stimulate or inhibit RNA polymerase. So these transcription factors can bind, I'm going to just do the stimulate one. So they bind to a promoter region on DNA. And well, they can stimulate or inhibit transcription. Because the entire point of this whole topic, this gene tech, is to muck about with transcription and translation. To express a gene, you need to be able to transcribe it into RNA. So that means messenger RNA specifically, by RNA polymerase. And then to translate it is going, letting it get to a ribosome and then, making a protein. That's the whole point of this topic. So this whole topic is how we can screw around with that and either make them transcribe more or less. So now it says, using all the information provided, evaluate the use of IPS cells to treat type 1 diabetes in humans. So again, the conclusion almost that you could say is IPS can treat type 1 diabetes in humans. Remember what I said before in the previous evaluate question? You look at the supportive statements, the contradicting statements, and then issues with validity. You should skip back to the previous question if you're watching it here and go find it. I can't remember what question number it is. But I was talking about this before. So let's look at the data to see what supports their use first. You can don't again go into saying other factors, this and that, blah, blah, blah. Go in and say what the specifics are. And what's interesting here is this was the first paper of the new spec series. And what I find interesting is that here there was nothing too much about people saying other factors or saying learned answers and memorizing mark schemes and stuff, right? Model answer rubbish. which I find interesting. But every single year after, they started to come up more and more and more, where people were like, other factors, or blah, 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 or like just saying a model answer completely irrelevant. So stop doing that. Like, stop. It don't work. And exam reports always talk rubbish on it. Here, it doesn't say very much about it. That's quite interesting there. And another thing as well, it says here, mice often became rats during the explanations, but this was not penalized, ! Which makes me laugh. It was fine, but just like, come on, man. What's going on? So again, we're looking at supportive statements. So what is supporting its use in treating diabetes in humans? Well, here, we can see that in this group A, which were treated with it, so we can see here that group A that were treated with it have a lower glucose, and it stayed low after. Almost as similar as C. So the supportive is that, you know, group A had type 1 diabetes plus the IPS. The glucose dropped to levels similar to mice without type 1 diabetes, which sounds pretty good, right? Is there anything else that's supportive? Not really. Now let's look at what's directly contradicting it. So this doesn't mean, oh, the other factors thing, as in what here is saying that it is not supporting it. So is there anything where it's showing it's going up, for example, where here, for example, it's fluctuating, isn't it? Sometimes at some points it goes higher. At some points it's higher than control, but again, it's similar, so it's not a hugely strong point to make, but it's just helpful to that. You're not going to lose anything by saying that, right? Now the issues with validity is what their problems are with their experiment. What's the problem? Like, for example, can I go and take this and apply it with every single mouse in the universe? Will I get the same results? Can any single scientist in the universe do this? And as well as that, here they're saying is can treat it in humans. Can I relate this entire thing and say this applies to humans as well? So here, already immediately I can say, well, no, there's a problem here that this has only been done in mice. Because that humans might behave different, their bodies might behave differently if we took their stems, their skin cells and did that, right? As well as that, it's only been done in 12 weeks. We don't know the long-term effects. Small sample size is not amazing because mice, we're doing this in preliminary research, right? Like here, like, it's not really relevant because 30 mice is a decent size for something as tiny as this. Bigger sample sizes up to the 100,000s, millions of things like trying a new drug out and stuff like that, really. The definition of phenotype then, so loads of people, just one mark, I think it was 90%, a lot of people just said the GCSE definition. What it is it's the expression of the genotype and its interactions with the environment. Not just, oh, it has this trait or physical thing or this or that, blah, blah, blah. So the inheritance of fruit color in summer squash plants is controlled by two genes, A and B. Each gene has two alleles, right? So it's important to appreciate that this is going to be a dihybrid thing that they're going to do if there's a cross coming up. So it says here, it shows the interaction of these two genes in controlling fruit color in the summer squash plants. Name the type of gene interaction shown in figure 7. So look at figure 7 and actually go look at what's going on. Loads of people said dihybrid and codominance. Dihybrid is nothing. All it means is it's just inheritance of two genes with different alleles. You could have monohybrid, trihybrid, all kinds of things like that. It's just them choosing to do two in one go instead. Codominance is where both of them express. So just have a look what's going on. So if we've got white, if we have little a, then it goes green. And if we have big B, it goes yellow. However, if you have white and you're inhibiting it, it can't become green, which means B can't act on it to become yellow no matter what. And if you have little b, then there's no functional enzyme, so it stays green. So this thing screwing up here, even if you have the right one for B, means you can't get to yellow. So the thing that's going on here is that this thing could mask the expression of this one, which is called epistasis. So epi means after, or upon, or on top. Stasis means stopping. So after stopping, something after it is stopping. It's literally in the name. For example, imagine this is you, you're nice, you've got lovely hair, and you're like so happy, and then here's you when you're bald. No hate to bald people, some people just like their hair, right? Now, imagine you have black hair, but you're bald. Even if you have the black hair allele, because you're bald, it does not matter whether it's black hair, blonde hair, blue hair, whatever, not blue, but like, you know what I mean? Like ginger hair, all of these different colors, does not matter because you are bald. The baldness gene is masking the expression of that. It's stopping whatever that is from being expressed, so it's epistasis. That's it. So it says, what fruit color would you expect for the following genotypes to have? So big A, little b, little b. So if we've got big A, it means we can't get to green. So if we can't get to green, then it can't go further, so that one's white already. Now little a, and big B. So if we're little a, we can get to green. If we've got big B, we're going to be able to get to yellow, so we have yellow. There you go. So now it says genes A and B are not linked. Complete the genetic diagram to show all the possible phenotypes, the ratio of the phenotypes expressed in the offsprings of this cross. So here then, a lot of people fumbled this quite weirdly for some reason. It's draw the cross and then just say what the phenotypes are, and a load of people just jumped into saying like 9331. Please stop doing that. Like, come on, man. What's going on? You can't just say, oh, it's dihybrid, 9331. Like, literally just can't. You need to go and check what they actually specifically are and relate it back to what the questions told you. So here then, if we're crossing these two, the gametes they're going to make, they're diploid, so we're going to make haploid gametes. One problem I see people do is they do this, and whatever, right? This is two separate, these are two genes, two different things. like that. So you know, if this was like AA, right, then you guys always know that you can just do this and this, right? We're inheriting 2 in one go. So what you've got to do is you've got to have both of the genes, one of each. So instead of it being like, for example, A and B, you're splitting into its genes. Into 2 each copy. Here it's AB, 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 because this is diploid, this is haploid. The meiosis makes haploid gametes. You're going to have half of this thing. In the same sense, if you have AA, you're going to have half of that there. That's why the gametes have 1. In this case, it's going to be half of these in each possible combination you can have. So the gametes, which I should have written here, whoops, the gametes are going to be big A, big B, or it could be big A and little b, or it could be little a and big B, or it could be little a and little b. Whoops, little a and little b, like that. Now when you're crossing it, just because I'm gonna do it up here, just because I want space. You're right, the gametes of one parent across the gametes of the other. So AB, 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 big A, big B, big A, little b, little a, big B, little a, little b. Cool. When you're crossing these, keep them consistent. So big A on the outside, little on the end. So big A, little a, big B, little b, big A, little a, big B, little b, big A, little a, big B, big B, big A, little a, big B, little b, like that, right? And then just keep them consistent. So big A, little b, little b, and then big A, little b, little b, big A, little b, little b, big A, little a, big little b, around that. So then it'd be little a, little b. Don't do big, little b, big B. Do big one first to keep it consistent so you can spot patterns easily. Little a, big B, little b, little a, big B, little b, little a, big B, little b, and then here little a, little b, and little a, little b, little b, little a, little b, little b, little b, little a, little b, little b, little a, little b, little b. So the genotypes of the offspring, now this is why you keep them consistent, because you can easily spot that they're just those, because those rows are all the same. So what you end up with is you've got little a, big A, little a, sorry, big B, little b, big A, little a, little b, little b, little a, big B, big little b, and then little a, little b, little b. Then phenotypes don't go, oh, 9331, and all this stuff, right? Go back and look at the question, because they've told you what's going on. If you have a big A, it's going to be white. So immediately, no matter what the B's are, you've got white, and this one's white as well. And then little a, big B, little a, lets you get to green. Big B means you can go to yellow. But then we have little a, which can get you to green, but then we have little b, which is yellow. Sorry, not yellow, green. Whoops, it stays at green. It can't go yellow. Whoops, it stays green. So the phenotypic ratio is going to be, well, white, and then one of these, and one of these. So instead of 9331 or all the garbage, it's going to be 2 that's white, 1 that's yellow, 1 that's green. So 2 to 1 to 1. So in this question, only 1 in 6 people, about 17% actually got the marks for this, because they just, it wasn't even the maths, it was just they misinterpreted the question horrendously, which is just like, come on, man, what's going on? A population of summer squash plants produce only green and yellow fruit. The percentage of the fruits that made, the ones that were yellow was 36%. So I'm going to write down the yellow, its proportion of them was 36% or 0.36. The total is 1. It says here, use the Hardy-Weinberg equation to calculate the percentage that were heterozygous for gene B. Heterozygous for gene B. So we need to figure out what's going to make yellow. So if you look at what makes yellow plants, in order to be yellow, well, go back here, sorry, just a sec. It was only green and yellow fruit. So it means if they're only green and yellow, then all of the stuff going on is to do with here. It can get to green, so by that it means it can at least get to yellow. So we're only dealing with stuff to do with bees, right? So if you're yellow, then you need to have the possibilities for yellow are big B, little b, or big B, right? And then the other option was green. To stay at green, the only possibilities is going to be little b, little b, little b, little b. Therefore, we know that P plus Q equals 1, right? And we also know that P squared plus 2PQ plus Q squared equals 1. These total are going to add up to 1 or 100%, so the greens that are there are going to be whatever's left over from 1 minus this. That means we have 0.64 of these that are green. Now, we've just said that green can only be BB, right? So if we said P to represent big B and Q to represent little p, that means that Q squared is going to equal 0.64, right? So then Q is going to be the square root of 0.64, which is 0.8. Now we know that P plus Q equals 1, so then that means P is going to be 1 minus 0.8, which is going to be 0.2. So now I know that P is 0.2, Q is 0.8. Now this equation says P squared, which we said was little p. Sorry, P was big B, sorry. So it's going to be BB plus 2PQ, which is going to be PQ is B little b. Whoops. B little b or little b big B. That's why it's 2PQ, because they're the same. And then Q squared, which is going to be little b, right? They're asking us for the heterozygous one, so we're looking for 2PQ. So 2 times P times Q is going to be 2 times 0.2 times 0.8, which is 0.32, but we're not done because they want a percentage, so that's going to be 32%. There you go. God, why is deleting stuff so annoying in this? So one way to detect and accurately measure the amount of RNA in a tissue sample, because we're looking for measuring RNA, is by doing RT-PCR, which is reverse transcriptase polymerase chain reaction. So what you do is you've got a reaction mixture with a sample for testing, and this is going to be, we're looking for RNA, so it's going to be an RNA sample, most likely. Reverse transcriptase, DNA nucleotides, primers, DNA polymerase, fluorescent dye. The method's shown here as well. So what you do is you have an RNA sample, and then what it does is you're going to, the amount of DNA amplified by PCR depends on how much RNA there was. The question's told you that it's reverse transcriptase PCR. So what's going to happen is this RNA is going to be, reverse transcriptased into DNA, and then it's going to be amplified by PCR. And then it's said here as well, the dye only fluoresces when it's bound to DNA, and then the intensity of the light fluorescence is increased as the PCR products accumulate. So overall, what you've got is RNA, which is becoming DNA using reverse transcriptase. That should always make sense of what's going on before you dive into the question. It should take you minutes to do, or seconds really. And then as well as that, you're making more copies of it using PCR. It's in the name. So it says, explain the role of reverse transcriptase in this RT-PCR. So a lot of people were saying that for some reason it makes RNA from DNA. Others were getting it confused with things like RNA polymerase, DNA polymerase restriction endonucleases. And a lot of people were saying hydrogen bonds between complementary base pairs and the formation of double-stranded DNA. It's literally none of that. What it does is it's going to make, so it's reverse transcriptase, it's making or producing complementary DNA from mRNA. What it does is it gets an mRNA, it lines up nucleotides, the DNA nucleotides, and it joins them together, reverse transcriptase. It's in the name. Because transcription makes DNA become mRNA, so if we're doing the reverse transcription, we're going from RNA to DNA. So it's in the name. Here it says, explain the role of DNA polymerase. The entire point of polymerase is it's joining adjacent nucleotides to form complementary DNA strands. Now, a lot of people were making mistakes. So, for example, they were saying it was joining, bases together, like complementary bases together, which is incorrect. What happens is it lines up the complementary bases. Imagine that's your template strand that we just made the complementary DNA. This is this. These nucleotides will line up like this because the complementary base pairings, because of hydrogen bonds. People were saying that DNA polymerase joins them side by side like that. You're not getting a strand, you just get little chunks of two things here, which doesn't make sense and is incorrect. It's adjoining the adjacent nucleotides to make a strand like that. And interestingly enough, it's like not that common in this paper, and this is the first one of this new cycle, but every single subsequent year, this error has come up more and more and more, which to me is suggesting that y'all are learning some stupid model answers and getting them wrong, or misunderstanding the model answers and memorizing the wrong thing. So please, it don't work. Any DNA in the sample is hydrolyzed by enzymes before the sample is added to the reaction mixture. Explain why. Well, what are they doing again? So they're getting a DNA, they're getting a sample, and they want RNA, and they're going to convert it into DNA, and then of that DNA in this mixture, they're PCRing it. If there was DNA in here to begin with, right, then it's not going to get really reverse transcriptase, right? But then it's going to get amplified, and you're just going to get more copies of it. So then the whole point of adding that the DNA there is to remove pre-existing, DNA, and then it's to, or otherwise this would be amplified. Many people were just saying it's to make the DNA single-stranded and all kinds of garbage like that, or to just say, whatever, like, oh, it was just done, but didn't explain why. Only 3% of people got both marks, and 80% of people got nothing. It's telling you in the question before you've added it to the mixture. So the question's told you that we have an RNA tissue sample. Before we've even added it, they've added something that's destroying DNA or hydrolyzing it. The only reason is to get rid of that pre-existing thing. Read the question, please. So here then it says the results from RT-PCR now to detect RNA in two different samples, A and B. Intensity of fluorescence. Remember in the question, which you should always refer back to, tells you that if you have more PCR products because it only binds to DNA, then that means you're going to have more fluorescence. The intensity of fluorescence is measuring the amount of DNA after PCR. I can guarantee you right now, if I look at this examiner report as I'm doing these questions, loads of people will have just completely ignored that. And then here we're looking at the number of cycles of PCR. Cool. And then the curves are showing A and B, and this is the RNA in two different samples, sample A, sample B. A quantitative comparison can be made by the amount of RNA in samples A and B. This involves determining the number of cycles reached to reach 50% maximum of DNA C. The amount of RNA can be measured as 1 over C. Use this information to calculate the ratio of RNA in A versus B. Use what the question's telling you, man. Lots of people were just saying utter garbage. Like, just because they just, like, what's going on? Like, here, the question's told you that if you reach 50% maximum, you get C. So I'm going to look for A and B. I'm going to look for its C here, right? So it's the number of cycles to get 50% of the maximum concentration. The maximum concentration is up here, which this straight line was just drawn horrendously. That's the maximum concentration. We want to find 50% of that. So that's 4.6, 4.7, 4.8. So we're looking for wherever half of 0.48 is, which is 0.24, which is here, right? We're looking at the number of cycles that hit these. So we're looking for you. Oh, we're looking for you. So if we do that, we end up getting, if the line will draw nicely. So here we got 19 for A. So C is 19 to get 50%. And then for B, so for B, it was 27 cycles, right? The question's then showing that the RNA can be written as 1 over C. So 1 over C is going to be 1 over 19. And for B, it's going to be 1 over 27. And it says the ratio in A to B of RNA. So A to B can be the same as 1 over 19 to 1 over 27. Now, if we're doing, generally, when you're doing a ratio of one thing to another, you're saying something to 1, right? So in this case, we're going to get here to from something to 1. So to go from here to here, you've divided by 1 over 27. So you're going to also divide this side by 1 over 27. If we do that, you get 1.42, which is roughly 1.4 to 1. There you go. So just one reason why DNA replication stops in the polymerase chain reaction. The polymerase chain reaction is where you add a bunch of nucleotides, you add some primers, and you add DNA polymerase, and the DNA polymerase will join these nucleotides together to make some strand. So if it's stopped, one of those things have gone wrong. The DNA polymerase is chilling. It's going to carry on. It's that you've run out of nucleotides or primers. Obviously, you would give one, but there you go. A lot of people just didn't say anything. They were talking about stop codon. They were talking about DNA helicase being denatured. Like, dude, it's asking you why this polymerase chain reaction stops. The only reason it would stop is because you've just run out of stuff to use. Scientists have used RT-PCR to detect the presence of different RNA viruses and respiratory diseases. The scientists produced a variety of primers for this procedure. Explain why. So the question saying RT-PCR, RNA in viruses, why do they need different primers? Primers are little short bits of base sequences that are complementary to the thing that you want to replicate, that you want to amplify. And what it does is it acts as a starting point, so then every single nucleotide joins like that. Alright, otherwise you could go the other way, or you could just join all kinds of random stuff together. So primers are literally saying, hey, mate, let's get started. So they're saying to use different primers for different RNA sequences, well, different complementary DNA sequences that would have come from that. So the only thing that's here, well, remember that they're using this, so they're going to reverse transcriptase this into DNA and then use the primers on that DNA they made. If they're using different primers, there's got to be different base sequences. So immediately, different base sequences must be present. Therefore, different primers are needed. Different, and then we've got to say complementary, because you need to explain why you need different ones. Primers are needed. That's literally it. A lot of other people were just saying, like, primers are complementary to different respiratory diseases, not saying what the specifics were, and a lot of them were saying that the viruses had DNA in them. No, it doesn't. What they've done is they've got this RNA, turned it into DNA using RT, and then they PCR'd that, and they're saying, why are you using different primers in the PCR of the DNA that came from RT, which came from RNA? So got to work back. What is the gene pool? Less than 50% of people got this, man. They were saying it's the amount of genes in a thing, this and that, blah, blah, blah. It's all of the alleles in a population. A population is just all of individuals that are the same species. If you're the same species, you're going to have the same genes. My hair gene is going to be the same as your hair gene. What's different is that I have different colored hair, maybe, because I have black hair. And the reason is because my alleles are different. Saying all the genes in a population is, you don't need the whole population for that. It's the number of all of the alleles, the different types of things that can come up in those genes in a population. Lord Howe Island in the Tasman Sea possesses 2 species of palm tree, which have arisen via sympatric speciation. These 2 species diverged from each other after the island was formed 6.5 million years ago. The flowering times of these 2 species are different. The flowering times. Using this information suggests how these 2 species of palm tree arose by sympatric speciation. 25% of people got no marks on this, and a lot of people were saying it's allopatric speciation without reading the dang question, literally telling you it's sympatric. Allopatric, allo means other, patric translates to fatherland. Other land speciation, right? Speciation is where you have so much evolution, you have so many changes in the frequencies of alleles that eventually you make 2 things that are 2 separate species, which mean they cannot interbreed to produce fertile offspring, because a species is something that can interbreed to make a fertile offspring. Sympatric, sim means same, patric means species. Patric means same land. So here it's sympatric, same land, speciation. So there are speciations are happening on the same land. Both types of speciation require reproductive isolation of some sort, which means that some population of them are unable to reproduce with the others, and as a result, this is where it starts to happen. Okay? So in sympatric speciation, in the same population this is happening, there's no geographical isolation. It's saying here why on earth the flowering times of these two are different and where it must have come from. So the same population, what must have happened is that a mutation arose, because mutations are random, arose to cause different flowering times, because that is the reproductive isolation. Flowering times, which is the reproductive isolation, because they're both reproducing at different times of the year. Therefore, there's no gene flow between the two of them. There's no exchange of alleles between the ones that are flowering at one point and another. So therefore, over time, what's happening is you're passing on different alleles, and you get a change in allele frequency. And then what happens is eventually, there's, you get different species that cannot interbreed to produce fertile offspring. In all of these scenarios, the reason that there is any variation is because of mutations. The reason I have a different color of hair to you is because of a mutation. Anything that's causing a genetic behavioral expression-y thingy difference is because of a mutation. So some mutation has happened where they're now flowering at different points, meaning they can't reproduce. That is the reproductive isolation. In allopatric, where you have another land, there's a geographical barrier stopping them from breeding with each other. So that's the reproductive isolation of this population versus this. Both of them hinge on there being reproductive isolation of some sort. In sympatric, there's something that's happening. For example, another thing could be like, for example, animals having a mutation that changes like their genitalia, for example, so they can't actually interbreed. So there's a mechanical problem there. Things like that. So here, I've seen some garbage strategies people have said of what they do to do these questions. Number one is make sense of just have vague idea of what the paragraph's saying. Don't just jump into the question and jump back. Understand what's happening. I think personally you should just skim through and read through the question just to know where bits and bobs of things are. And then as well as that, have an idea of what they're describing. Don't go in and think, oh, what topic is this on? No, that's not the point. The point is the question will tell you something. Go back to the paragraph, refer to it, and explain it based on your own knowledge. So I'm going to skip out me reading it just because it doesn't make sense for me to include that in the video, but you can pause the video and have a read now. Alright, so then it says, assuming no one with AD died in 2014, calculate the annual percentage increase in AD cases for America in 2014. So we're looking at the percentage increase for 2014 of cases, right? Annually. So go back, so when it says lines 2 to 4, it doesn't mean what you need is in lines 2 to 4. It's telling you where to go and find the bit they're referencing. You need to read around it and before and after it to see what you need. Because if I look only at lines 2 to 2 to 4, maybe then the answer that I don't need is there. So if we look at line 2 to 4, at the start of 2014, the number of Americans was this. Every 30 seconds, another person develops AD, right? Like, if you had just looked at this singular line there, you would have got literally nothing from that. So the question says, it's a non-reversible brain disorder. At the start of 2014, the number was 5.4 million. So at start, it was, whoops, I've forgotten the number already. 5.4 million, 5.4 times 10 to the 6 cases. And the question says, every 30 seconds, another person's developing Alzheimer's disease. So that means we're going to have to work out how many of there are in a day, right? So what I mean is every 30 seconds, new case. So we need to work out how many seconds are in an entire year. So 1 times 365 is how many days, times 24 is how many hours, times 60 is how many minutes, times 60 is how many seconds. Because 60 seconds makes an hour, sorry, a minute, which makes an hour, and 24 hours makes a day, and there's 365 days in a year. So we need to work that out first to find out the total time in seconds. If we do that, we get 31536,000. So it says every 30 seconds you get a new case. So you're going to divide this answer, this time in years, divided by 30. Because every 30 seconds you get a new one. How many 30s can you fit in there? So if we do that, we get 1051200, right? So that means these are new cases. So at the start, there was 5.4 times 10 to the 6. At the end, there was this plus 5.4 times 10 to the 6 plus the 1051200, which is going to equal, if I just do that now, it's going to be 6451200 at the end, right? The question's saying the annual percentage increase, this is across 1 year, so you would do this end minus the start over the start. So 6451200 minus 5.4 times 10 to the 6 divided by the start, which was 5.4 times 10 to the 6 times 100. If we do that, we get, that gives us 19.46, which is roughly 19.5%. Next it says, explain how donepezil can improve communication between nerve cells 7 and 9. So let's go look at lines 7 and 9. So 6, 7. So if you look at lines 7 to 9, so 7. So some of the symptoms associated with AD are associated with communications between the nerve cells and reduced by taking donepezil. Right, if you just stop there and we're like, oh, I can't find any answers. Donepezil inhibits the enzyme acetylcholinesterase. If we look back, it's only telling you to go look at 7 to 9, not that the answer is there, right? So if you go back just a little bit, in the brain of a person with AD, there is a lower concentration of acetylcholine, which affects the communication of nerve cells and causes memory loss and confusion. Lots of people, what I just see is that they just jump straight to 7 and 9, like, I can't find the answer, and then just yap about garbage things they think a model answer is. Here, the problem, they've told you, is that there's less, less ACH, so there's less communication, which leads to symptoms. But the question's just told you as well that donepezil inhibits ACH. Your job is to explain how this can improve communication between nerve cells. So if your donepezil inhibits ACH, acetylcholinesterase, then it means you're going to have less ACH breakdown. Therefore, more ACH will attach or bind to postsynaptic membrane receptors, and then therefore you get increased Na plus influx, which leads to depolarization, which if you hit the generator potential and you get your threshold and this and that, blah, 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 then you get the action potential. Lots of people didn't even talk about the postsynaptic neuron. They were just like, there's more here. Okay? Like, you need to describe from start to finish the communication between nerve cells. The nerve cells communicate with releasing a neurotransmitter, binds to a receptor, and then it opens Na plus ion channels to cause depolarization. Donepezil, to this. So come on. So now it says, explain 2 reasons why there's a high frequency of this mutation in Yaramul. So if we look at this here then, so 13 to 15, right? So Yaramul is a town historically isolated in the Andes Mountains. The population of this has the highest frequency of this mutation in the world, right? Let's have a quick look and maybe just read around it because it's not going to be there. The origin of this has been traced back to a common ancestor in the 17th century. Natural selection has not reduced this in the population. So what's happening here then is that we've got this population here on its own, and there's a lot of this E2AT here. Right, of this mutation. There's no populations coming in, and there's no populations coming out. Oh, well, there might be, but not very many, so there's not, like, a lot more variation. So some people might actually have another mutation that stops it from happening, this and that. So what's happening is they must all just be inbreeding, like, with themselves, or there's a small gene pool, which means that the allele is still inherited from generation to generation. You can't say founder effect, because no one's come here and done that. It's like, they've just been living here, it's asking that. So here, they are isolated. Therefore, you have a small gene pool, so a small variety in different alleles, or you could say inbreeding. As well as that, it means you've got to describe what's happening. The allele is inherited through generations from the common ancestor. You know the bit where it says here the common ancestor had it and there was no natural selection? They all got it from there. It's saying explain two reasons why there's a high frequency and why it's there. So it's because it's from that common ancestor through generations. There was no selection against it. There was no, there was nothing that would make them less likely to survive and reproduce, blah, blah, blah. A lot of people were talking about genetic bottlenecks, the founder effect, and all of that stuff in isolated areas. It's not correct because, what? And then a lot of people were saying that the late onset of it could allow them to reproduce and pass on the mutation. It would explain why the mutation, had not been reduced rather than why there was one high to begin with, if that makes sense. Right? A lot of people just said that there was a random mutation rather than it being from a common ancestor. There was a common ancestor here. It was living here, this small pool. So because of that, they're interbreeding with each other and they all the population inherited it all from that one original geezer that was here from a long time ago. Explain why natural selection has not caused this frequency of this mutation to drop. So again, thank God there is no model answer garbage here of rote learned answers, but in subsequent years I've seen them come up. So I can tell that this, because again it was the first year, a lot of people have just been trying to hack the system, memorizing stupid answers that just don't work. Stop doing that. Here it says, this natural selection has not reduced this allele in this population, line 16 to 17. And the question tells you that natural selection has not reduced this, and that's it. There's nothing else here. So it's up to you to explain what's going on here, right? Well, look at the whole context. What's the point of that allele? So it says here, a gene mutation on this causes it to develop at, a mean age of 49 years. So if this mutation makes you get it at 49 years old and there's no natural selection, they're old enough to have reproduced. And that's what it is. That's why you can't just jump to the line and say, I don't know what to say, bro. You've got to use the whole question. So 80 develops with this mutation at 49. They've already reproduced if they have wanted to. There's nothing stopping them from, surviving, reproducing, blah, 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 and all of that jazz, because, like, they're still able to reproduce. A lot, you know, we still have illnesses and problems today, and no people are dying and not being able to survive and reproduce. Well, very few are, because they're able to actually survive and reproduce. The age at which this mutation causes can vary, so just one explanation for this. So if we look back at lines 11 to 12, where are we? So it says here, the age at which this can vary. So the general rule of thumb is if you have a gene, a singular gene, in theory, it should, or an allele really of a gene, it should be expressed or not. Here, if it's expressed and it's giving you an age of 49, it means it's going to be, here you go, let's go. But for some reason, there's something coming in the way where it could be a bit later, a bit less, you know, a bit more, and a bit less. So a lot of people were talking about exposure to mutagenic agents, levels, different levels of acetylcholine, which I guess is kind of fair, but like the mutagenic agents, nah, not really, because they already had it to begin with. They already have it. What it's saying is that people who already have this tend to have it at either 45, 48, 49, 50, and so forth. What it's trying to get at here is there's something stopping it from being expressed, which is epigenetics. So epigenetic control due to an environment. So for example, like, you know, due to environmental factors like, I don't know, like, you know, diet, exercise, smoking, etc. And it's because of either increased methylation of genes, reducing its expression until, you know, the time comes along, or decreased acetylation. That's what it's trying to get at. So here it says one scientific study analyzed chromosome 14 and involved 102 people. So again, 102 people looking at chromosome 14, and they recorded a sample size of 204. The reason they got 204 is because you have 2 copies of the 14. So if you have 102 people, they're going to have 204 of these samples, 204 chromosomes they're tested. They found 75 E to 8T mutations, but only 74 AD cases. Suggest explanations for the figures that they recorded. So let's just have a look if there's anything here that might be useful. So if we look here, blah, 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 they analyzed these and this and that, they recorded sample size that, detected that, but only 74% cases. They identify individuals with mutations by whole genome sequences. They decided a DNA probe would be a suitable method. Fine, so it's not very helpful here. So we need to go and have a look at what's going on. So if we just one sec again. There is a change in the triplet to GAA to GCA. Fine. So back here then, what might be the reason as to why? So we have 204 chromosomes, because remember each person has 14 and a 14. They found 75 E2ATA mutations, right? And they've told you in the question that if this is dominant. So if it's dominant, it means it should be expressed. So that means that the 75 that they found should have E2ATA, but for some reason there was only 74 cases. You only need 1 copy for this to express. Somebody might have had 2. So they, you know, had 2 of these E2 whatever, blah, 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 E2, blah, 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 right? So then all the other ones might have had 1. If they all had 2, you'd have half of that. So that's what it must have been. It must have been that one person was homozygous dominant and had 2, you know, of these alleles. Because remember, that's why they had 14 individuals, 102 individuals, 14 chromosome 14s, right? But you're going to get 204 of them because there's 2 copies of them. It says, suggest why a DNA probe was not considered suitable for detection of this mutation. So if you look at 22 to 23, what's going on here? So the scientists identified individuals with this whole genome sequences. They found that a DNA probe would not be suitable for this. The mutation is from GAA to GCA. So the question's asking you why DNA probe for GAA to GCA is not good, right? A DNA probe is where you've got some fluorescent dye or whatever, a radioactive thing attached to a bunch of base sequences that are complementary to what you're looking for. And then it'll bind there, and then, you know, you will look at it under like UV light or whatever, and then you'll see what it's up. So a lot of people just fumbled on this. 10 people didn't even try the question. 5% just skipped over. And as well as that, only 25% actually got a single mark on this question. So, you know, all of the people were just saying you couldn't determine whether the mutation was there or not. A lot of people were saying that, like, you'd need a lot of different primers and all kinds of just stuff like that. Look at it again. GCA, dude. GCA is 3. You're going to have that all over the place. GCA is very common. You don't need to, like, have that knowledge in your head. 3 bases that come like that. It's probably going to be a huge amount of those present. So you're probably going to get false positives, or, you know, if it was positive, if you know it was positive, you don't know if it was due to the allele. Because I could, you know, you could look anywhere in anybody's body and they're probably going to have GCAs there. So I hope that helps. Please like, share, and subscribe. You know the drill. And also for biology and actually all A levels, model answers don't work. Rote learning things just does not work. I'm sorry, I don't care who's telling you that it does. It just objectively doesn't work. If you know your stuff and understand what's going on and you look at the question and answer it directly, you will end up saying the model answer. The only time model answers ever work is if when you are actively just recalling something and you're recalling the right thing. So for example, if a question says, I don't know, describe ventilation in humans, you would say that the external intercostal muscles and the diaphragm muscles contract. This increases the volume of the thorax, decreases its pressure, and then that causes air to move down a pressure gradient into the thorax and then into the lungs. Fine, that's your model answer, but like, you need to understand it anyway to say all of that. You know, there's no point memorizing that, because if a question tests you on a piece of it and you need to recall it, you're useless and just don't know what you're doing. Go and read the exam reports when you get a chance from this year onwards. Every single year it has model answers coming up more and more and more with rote learned garbage being spewed in exams. It doesn't work, and when I read it, it makes me want to die a little bit inside. Stop doing that, please. Read the question, answer what the question's saying, and also the model answers you're learning sometimes are wrong. Like, some of them are objectively wrong, so stop doing that. Thank you very much, and you know the drill. Like, share, and subscribe, and yeah, see ya.